Welcome to the CannMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I am your host, Ben Amaralt. I'm the marketing manager at Medicinal Genomics and proud member of the team who puts on the CannMed conference. A few housekeeping items before we introduce this episode's guest. Check out CannMedEvents.com for all the latest information about the CannMed 24 Innovation and Investment Summit, happening this May in Marco Island, Florida. CannMed 24 is an all-inclusive event featuring world-class content, 4-star accommodations, ample networking opportunities, and meals and drinks included. Be sure to sign up for email alerts at CannMedEvents.com so you don't miss any announcements leading up to this year's event. If you plan to join us in Florida, I encourage you to buy your tickets now. Space is limited this year and rooms are filling up. And if you can't join us this year, you can still stay connected with the CannMed community by following us on social media. We're on Instagram, Facebook, X, and LinkedIn. Just search for CannMed Events. And you can also enjoy free access to all the podcast archive episodes, as well as videos of all the past CannMed presentations in our CannMed video archive at CannMedEvents.com. Our guest this episode is Tim Lefever. Tim is the Director of Discovery at Nalu Bio. He's an experienced director with a demonstrated history of 20-plus years of preclinical in vivo research and leading effective teams at organizations such as Canopy Growth and Research Triangle Institute. He has authored or co-authored numerous manuscripts and presented research findings at a variety of scientific conferences over the past 20 years. At CannMed 24, Tim will present Characterization of Less Studied Cannabis Phytochemicals, which investigates the pharmacological, physicochemical, and basic toxicological characteristics of cannabis phytochemicals. During our conversation, we discuss how Tim and his team choose which phytochemicals to study, what makes the varin cannabinoids particularly interesting, the difference between chemically derived compounds and plant-derived compounds, which phytochemicals show the most promise for therapeutics, and modifying phytochemicals to improve their effectiveness. Before we get to my conversation with Tim, I'd like to thank this episode's sponsor, Brains Bioceutical. Brains Bioceutical is a global leader in EU GMP certified production of naturally sourced active pharmaceutical ingredients for the pharmaceutical and nutraceutical industries. With a unique suite of licenses, Brains Bioceutical is one of the only natural plant-based phytochemical API manufacturers in commercial production today and is involved in academic and clinical trials across the globe. Learn more at brainsbioceutical.com. Okay, and without any further ado, please enjoy my conversation with Tim Lefever. Good morning, Tim. Thanks for joining us on the podcast.
Thanks for having me, Ben. I'm excited to talk to you today and excited for the upcoming conference, the CannMed conference in Florida in a couple weeks.
Yeah, I know it's coming soon. We're definitely excited. And I'm excited to talk with you today about the work that you've done to investigate some of the lesser known compounds in cannabis. But before we kind of get into which compounds and cannabinoids you studied and the results that you found, I wanted to ask what specifically you were investigating. Maybe more so, what basic questions were you looking to answer with this work?
Sure. So, as most folks in this space are aware, the cannabis plant is incredibly complex. There's a number of different cannabinoids, flavonoids, polyphenols, and well, some of them are fairly well researched. There's many others that have just a smattering of information here and there. And so what we wanted to do was take a look at some of these less studied, a little bit more rare, or sometimes called minor cannabinoids, and just get some basic information off of them that can lead us in directions where we can decide if we want to potentially commercially pursue them for health benefits or use them as a scaffold in our drug development program to use medicinal chemistry to optimize them further for therapeutic potential. And so with this work, we chose a number of cannabinoids. We looked at both the delta 8 and the delta 9 isomers of THCV, as well as CBDV, CBGV, CBCV, and CBC, and canflavin A. There was a lot. I had to think about it for a second. And again, some of these have much more data in the literature than others. And so the basic information was really around ADME, PK data, some physicochemical properties of these cannabinoids, a little bit of pharmacology work where we chose some selected receptors or ion channels, probing for their activity at those. And again, just to get a basic sense of where these stand so that we can follow up with more comprehensive in vivo biology studies where we can really nail down efficacy and safety.
Great. No, and I was hoping you might help me, or help me and I guess the audience, to sort of define a few of these terms. So, looking through your abstract and what you're investigating with these different compounds, there's pharmacology, physiochemistry, and toxicology. So I was hoping you could kind of define what are those terms? Like what are you really looking at? So maybe start with pharmacology.
Sure. So pharmacology is really just how a chemical molecule or some entity interacts with our system, most often by binding to and activating or having some effect on a receptor or, again, an ion channel. Lots of different ways you can approach that, but to boil it down, it's basically how does the chemical do what it does in the body. Physicochemistry is basically just the chemical properties of the compound, right? So, everything from as basic as its molecular weight to, say, solubility. So in various solvents or phosphate buffered saline, how soluble is this compound, which is related to how much we could potentially mix or dose easily, which is related to kind of the ability to commercialize it, or again identify a characteristic that we might be able to improve upon. And toxicology is pretty straightforward. Obviously, it's just really how safe are these, and there's many ways to approach assessing toxicology of a chemical or a molecule. In this case, we decided to go with a C. elegans model that we had some familiarity with some previous work that we found to be a really good early place to start that gives us a good sign of both some acute and subchronic toxicity that we can screen fairly rapidly compared to a more traditional in vivo rodent model. But that, again, gives us an early indication of this is one we need to do some more work on, or this is one right now we just don't think it's one we want to pursue.
Okay, great. Thanks for that.
Mm-hmm.
And so in looking at this in the compounds that you studied, were there any surprises or sort of results that you weren't expecting?
Yes and no. So there were definitely a few things, and this was, again, one of the goals. Some of these we looked at purely from a scientific interest perspective. So, for example, the delta 8 and the delta 9 THCV isomers. If you look across the marketplace right now, a lot of the THCV isolate products contain a ratio of both isomers in every direction and permutation you can think of. So we wanted to understand a little bit more about each separately, and what are the pros and cons or the benefits of having more delta 8 versus delta 9 or vice versa. And so while they really, you know, are very similar, much in the same way that delta 8 THC and delta 9 THC only differ by the location of the double bond on the carbon, right? That's molecularly, that's the only difference. But when we did some of our binding and functional assays, we found that delta 8 seemed to be a little bit stronger antagonist at CB1 compared to delta 9 THCV, and then the exact opposite for the CB2 receptor. Delta 9 THCV was a little bit stronger antagonist than delta 8. So again, the implications there, once you start to understand some of this pharmacology, you can either specifically generate a ratio of the two isomers to drive a tailored effect, or isolate and purify just one isomer, and get the most bang for your buck, if you will.
Okay, so you were investigating delta 8 THCV and delta 9 THCV.
Yes.
And there are a lot of V's when you're kind of rattling off all the different compounds. So, and I know that's the varin variety. I don't know what the term is there, but so what specifically was interesting about varins that, you know, there are so many of them in this list?
Well, so the varins are a little bit interesting to us, right? And again, the difference between the varins and the non-varin cannabinoid acronyms that you're familiar with is, right, again, just the number of carbons in the alkyl tail, right? So the varins have 3. So CBD V has 3, CBD has 5, right? And so for a while, and I think it still persists a little bit today, folks tend to look at the number of carbons in that tail when they talk about agonist activity at CB1. So typically kind of an indicator of how intoxicating a cannabinoid may be. We don't pursue anything intoxicating, but in this case, we see that the varins are slightly more pharmacologically promiscuous, and are a little bit more like CBD in that regard. They don't take a hammer to really any one particular target or receptor or site, but seem to kind of have this general activity across several systems that both contribute to health benefits or therapeutic potential, but also likely safety, as we see with CBD.
Interesting. So in doing research for the conversation today, I was checking out your website at Nalu Bio, and there's a lot of talk about having chemistry-derived compounds versus hemp-derived compounds. So in doing this work, which were you using? Were you using hemp-derived or chemically derived?
So these were all synthetic, chemically derived compounds, which is our platform at Nalu Bio. So we believe that the synthetic mimics of phytocannabinoids that you can extract from the plant or that are present in processed cannabis material offer some advantages, and so that's why we've decided to go down that route. We understand that there's a lot of negativity around the word synthetic, especially in this space, and especially after the years of the K2 and spice issues that we all are very well aware of. But we're hoping that we can kind of get over that stigma and embrace what the synthetic approach has to offer from a both consistency standpoint, from batch to batch consistency, which is achievable with plant-derived extracts for sure. But also that we're able to explore some of these more minor or rare cannabinoids that at this point the genetics don't exist, or they're in process to make extracting from the plant not very feasible, or not sustainable, certainly from an environmental standpoint.
Yeah, so what kind of levels are we talking about? Like some of these compounds, what is the typical amount that the plant produces, and what were the levels that you were using in your investigation?
Right. So I'd have to look to get really accurate answers for you, but most of these very, very small amounts. I know CBDV, there are some varieties from Africa that produce, I think, 4 or 5%, maybe a little bit more. I don't know if GW has done some of that work as well, but very small amounts. Nothing like Delta 9 THC or CBD at this point. Although I will say that Phylos has recently put out a press release that they have achieved genetics for a very high Delta 9 THCV plant that they're growing and extracting from.
So I wonder if you could explain a little bit about how are you kind of deriving these chemically? Is it similar? I know that, for example, with Delta 8 THC, it's kind of using the CBD molecule and treating it with acid to sort of convert it into Delta 8 THC. Is it a similar process that you're using now where you're using a kind of cannabinoid as a base and then transforming it?
In some cases, yes. Ultimately, how we approach the synthesis for any cannabinoid is tailored specifically to that cannabinoid. So as we begin to explore what's the best route, we look at various input materials, what's going to give us the best yield, the best purity, and then as we begin to hone in on what's that optimized synthesis, we further tweak it as we need to go from bench scale up to, say, kilogram scale synthesis work, and there's some more tweaks in there. So the very long answer is some rely on other cannabinoids as input materials. Others use other input materials like divarenol or olivitol, which are very common in this space. But each one is different and specific.
Right. And now, of the phytochemicals you studied, which have the most promise and why?
That's a very interesting question. So with these basic studies, it's a little bit of a tease. So you're just kind of scratching the surface enough to get very excited about a few of them for a few different reasons. And others you kind of are left scratching your head that you thought going into it, well, I really thought we might see something there. So across the board, more work is going to be done. But I can say that with the work we just completed, in my opinion, based on the data we have, I would think CBGV and canflavin A are particularly interesting. Canflavin A has been published on a little bit over the years. It occurs, minute amounts in the plant, which has hampered it to some extent. It hampered its research to some extent. But it has fairly profound anti-inflammatory properties, and as a polyphenol, it's pretty interesting to explore that space. It's a little bit of a departure from say the traditional cannabinoids we talk about. And in the case of CBGV, this is one, again, there's limited information out there about it, but it set itself apart from the pack on a couple of key ADME metrics like half-life and microsomal stability that looked a lot more like what you see in typical therapeutics in the marketplace today, which was a very nice surprise for us. We did not expect that.
Oh, so explain that a little bit more. So what specifically?
Yeah, so with CBGV, so we did several different solubility assays. As working with cannabinoids, a common issue is that they're not very soluble. And so we wanted to understand where these fall in relation to some standards. So we did some kinetic solubility, some thermodynamic solubility, and across all of these, what you see is that you can achieve the highest concentration with CBGV compared to all of the other phytochemicals we tested in those assays, which just basically means it's more soluble than most, which is a very good thing. And on
In what way?
metabolic, well, just that you can, it's easier to mix and get into higher concentrations in, say, a beverage or an oil, or a format where you can deliver more of the compound without it precipitating out, or you can maintain homogeneity of your finished good, so that when someone is taking it, you can potentially deliver less compound and achieve the same effect.
Got it. So it's less so sort of its interaction with the body, but it's more of a manufacturing problem that it's solving for. It's going to give you a more consistent product.
It's a little bit of both, actually. And so on the microsomal stability and the half-life work that we did, which is basically you're looking at how the molecule is moving across different microsomes, in this case, liver microsomes. And so what we saw is that CBGV had the greatest half-life, so more of it was sticking around, which speaks more again to its metabolic profile, but also that it had a better transporter profile. It was able to move more freely than some of these other cannabinoids, which again is just it caught us by surprise, and certainly now gives us motivation to continue following up with some in vivo work.
Right. And I'm glad you brought up that term, because I wanted to talk about sort of the difference between, in vitro and in vivo. And I know you can't really predict the future, but when moving from in vitro to in vivo, how can sort of these results, or like you said, you get excited about a molecule, how can then, when you translate that into in vivo, how can things change?
It's usually, I'd say the translatability from in vitro to in vivo is pretty high, depending on the data you're using to inform the other, right? So if the in vitro data, say, is in vitro biology data about, say, the pharmacology, so the binding and the functional assays we ran. So if you get a good understanding of this molecule has this activity at this receptor, and this receptor mediates these effects or interactions, you can follow that up in vivo, and typically you can see, right? So for example, if you see CB1 agonism, right, you would say, okay, now we're going to put this into an in vivo model, and subsequently we'll see analgesia, right? That's a common delta 9 THC effect in rodents, or any CB1 agonist with really any decent efficacy.
Right. So when you were talking about CBGV, so based on the pharmacology there, like, do you have any idea like what sort of conditions that might be helpful for?
It's a little bit tricky on the pharmacology side of things. We have to do a little bit more work there, but in some of the in vivo work that we've done, that I hope to be able to have some more data to present in a few weeks, it seems to be very stimulating, which is also interesting. So we're seeing a dose effect there, which in the model we used can sometimes be predictive of neuroprotective effects, which can come into play when you're looking at, say, traumatic brain injury or Alzheimer's. There's lots of different ways to approach those, and they're very complex studies, but I'm pretty excited at the chance to explore CBGV further, in some of these models, and really start to understand what we've got on our hands.
Interesting. And now, have you done work to investigate sort of combining these different compounds to see what the effects are? I mean, I know that the entourage effect is real.
We haven't yet. So our approach at this point is really trying to screen these minor or rare cannabinoids, through our platform, and get an idea, or rank them really by those that we feel have the data behind them to pursue further for either a commercial pipeline or to use as a scaffold for our therapeutic program. But at this point, we haven't combined any. We think we're hoping that we're adding value by adding to the knowledge we know about these individual cannabinoids. And well, I don't think we'll ever be able to recreate the chemical complexity that leads to the entourage effect. I think our goal is that if we understand enough about these cannabinoids or other phytochemicals individually, we can start to thoughtfully put them together with other phytochemicals or with existing, bioactive nutraceuticals or health supplements to really bolster specific effects, while not having to worry about the myriad of other chemicals that may be in the product.
Right. No, and that was something that I noticed when I was looking at the website as well, was sort of this idea of sort of combining the compounds, the cannabinoids, with other vitamins or probiotics, proteins, things like that. So that's something that you're investigating?
Yeah. So the commercial side of the business is actively exploring existing combination supplements and how the introduction or the addition of a cannabinoid or more than one cannabinoid may further enhance their effectiveness. So we've identified several that have a very solid clinical literature space behind them. They're very solid as far as efficacy is concerned. From a safety profile, they look good. And so we're now taking what we've learned about some cannabinoids and putting those in there. And eventually, once we start getting these manufactured, the intent is to do some small clinical trials or focus groups to understand their efficacy in humans.
Excellent. And now, which compounds are you looking to kind of move into the next phase of your work?
Definitely, definitely canflavin A and CBGB, as I mentioned. It's a little early on those to be involved in the combination products we're just talking about. But we definitely want to understand a little bit more about those. And as I said, if there's potential for them on their own, that's one thing. But the really interesting part about the Nalu kind of ecosystem is that we have this commercial side where we're developing the synthetic approaches for these phytocannabinoid mimics, but that also informs us on the therapeutic side. So our drug development side, where we can take what we know about existing phytocannabinoids or phytochemicals and do some of this work that we're talking about today, and then say, that has a lot of therapeutic potential, but it's not very drug-like. It's not very soluble, or it's rapidly metabolized. So how can we use medicinal chemistry to modify that molecule and improve upon those characteristics, while also either retaining or improving the pharmacological activity for the indication or the endpoint that we're looking at? So really, it's this, like I say, it's kind of this feedback loop ecosystem we have, where no matter what data we're collecting on either side of the business, we're learning more and more and more about these different structure activity relationships and how we can take that knowledge to inform really either side of our business.
So that's interesting. So, for example, when you were talking before about CBGV and it being soluble and that being a good characteristic, are you sort of saying that you can look at the structure of that molecule and maybe apply some of that to maybe another molecule that isn't as soluble to try to give it that ability?
You could. Typically what we're trying to do, and what a lot of folks do with structure activity relationships, is by looking at the properties of a number of molecules, so a large sample size, you could start to see patterns emerge where then you can more confidently say, okay, so making a modification here seems to be critical for X.
Hmm.
And so when you get large enough data sets, you can start to tease those things apart, and then absolutely you can start to tinker, molecularly tinker, with purpose. And so we have the first phase of anything we do at Nalu is our in silico platform. So we have several different modeling capabilities where we will put some of these rare minor cannabinoids. We also use it for novel chemical entities that we develop for the therapeutic side. And so we're able to model how these might interact with different biological systems, from metabolism to PK, as well as their pharmacology. So how they interact with certain receptors in the brain. And so that's kind of our first pass. And when we see favorable results come out of that work, that's when we move into the type of work we're talking about now. So we try to confirm that the data we've modeled using, PChem work, ADME work, in vitro studies. And so that's step two. And if we continue to see favorable results, we keep investigating them until we have a very, very strong profile and rationale for commercialization or therapeutic work.
And about how long does that whole process take?
It depends. It depends on if we have the target validated from say molecular docking perspective. It's not too bad. If we have to validate a new target, that can take us, a couple weeks maybe, depending on the complexity and, depending on if we're looking at, say, the orthosteric sites versus allosteric sites, what kind of training sets are already available. But it's pretty amazing how much this has evolved in just the last few years, especially with machine learning and AI. It's evolving very, very quickly and becoming an incredibly useful tool, that I think most biotechs and pharma companies are becoming actively reliant on. And it's certainly been working very well for us. And again, any data we collect, anything we collect, whether it's our data or something we find in the literature, we can put that data back into our system, and it's going to refine its predictions going on, so that we get more accuracy, better confidence in what our models are predicting.
And that's the in silico work that you're talking about?
Mm-hmm. Mm-hmm.
So even beyond that, so you have the in silico, in vitro, in vivo, and all that. How about that whole process? How long does that all take?
Yeah. So on the commercial side, so if we're just trying to understand a rare, say, CBGV, for example, if we wanted to work that through to the point we have enough confidence to start exploring how to synthesize it and optimize that synthesis, we could maybe 6 months to a year before we really have a good understanding, maybe a little bit longer, if we really wanted to explore a couple different endpoints. On the therapeutic side, obviously that's the drug development pipeline is a much longer game, and the stakes are a little bit different, right? So if we were to use, again, just CBGV, for example, as a scaffold, and using our computer-aided drug design, to say generate, 500, 600 novel chemical entities based off of CBGV, then running those through, say we get a few that look okay, we may do another round of computer-aided drug design or medicinal chemistry to further refine those NCEs into potential hits, that then we start to, again, verify, use physically in these very basic in vitro assays. And as you know, then from there it's more refinement and more work, and until you eventually get a lead candidate that you really want to push forward, and hope that you get into first in human studies and clinical work. But that that probably a couple years, at best.
Right.
You might get lucky, but yeah, we're moving along at a pretty good clip for our size. But yeah, that's, if somebody figures out how to speed that process up, they'll make a lot of money.
Right. And so the goal is, if I'm hearing you correctly, is to actually find a novel compound that sort of mimics the action of the natural compound and then commercialize that.
Correct. Or we enhance that activity, right? So we can explore ways to, say, change we can try to make modifications and maybe say, add bias to these molecules, or which signaling pathway they may prefer over another, knowing that activating one pathway may have common side effects that we want to avoid. And so that's just an example of one way that we can keep working on this, and again, get to hopefully a very safe and effective therapeutic, or a library that we can begin to use structure activity relationship work to really try to find the key components that we can exploit.
Excellent. All right, Tim. Well, winding down here, before I let you go, I wanted to give you an opportunity to share any other resources that you think folks might be interested in reading more about, if they're interested in this topic. And then also any information about you and Nalu Bio that you'd like to share, please do so. And I'll be happy to put any links in the show description so people can find it easy.
Yeah, I think I don't have any specific resources. You can check out our website and learn a little bit more about what we're trying to do at Nalu, and our approach to synthetic cannabinoids, whether it be synthetic mimics of phytos, or our therapeutic pipeline. Yeah, I can't really think of any other resources to send folks to. I will say that the majority of this data, if not all of it, will begin packaging up, and we'll hopefully get out in a peer-reviewed publication here before the end of the year, and hope to have many other publications following.
Excellent. Yeah, and in the meantime, if you want to have a conversation with Tim and the folks at Nalu Bio, make sure you come out to CannMed and pick his brain.
Yeah, definitely looking forward to it. It's going to be a great conference.
Yes, we are very much looking forward to it as well. All right, Tim. Well, thanks again for the time, and can't wait to see you down in Florida.
Thank you, Ben.
Hope you enjoyed my conversation with Tim Lefever. Check out the links in the show description to learn more about the topics we discussed. And thanks again to this episode's sponsor, Brains Bioceutical. In the meantime, go to CannMedEvents.com to check out all the details related to CannMed 24. Speakers, poster presenters, networking events, panels, workshops, meals, and more. All the information is there. While you're at CannMedEvents.com, sign up for email alerts so you don't miss any announcements before the event. If you are planning to join us in Florida this May, register for your ticket package now. Space is limited and tickets are going fast. I also invite you to follow us on social media. We are on LinkedIn, Instagram, X, and Facebook. Just search for CannMed Events. And lastly, please rate, review, and subscribe so you don't miss an episode. All right, that's it from us. Stay safe, stay healthy, and be sure to join us for the next CannMed Coffee Talk.